Photophosphorylation, mechanism of photophosphorylation in light of the modern concept as well as in view of the earlier concept. We'll discuss its types, and first of all, let's understand what we mean by photophosphorylation. Definition of the photophosphorylation, it simply means the synthesis of ATP due to coupling of ADP with inorganic phosphate in presence of an enzyme. That enzyme is ATP synthase, and of course in presence of sunlight. So in simple terms, we can say it is the synthesis of ATP in presence of sunlight. Photophosphorylation, that's coupling of ADP with inorganic phosphate in presence of light. It was first discovered by Daniel Electron in 1954, and Daniel Electron is an American plant physiologist whose research led to greater insights into the operation of photosynthesis. He discovered photosynthetic phosphorylation, which is a process by which chloroplasts use sunlight to generate ATP. That means you have an ADP, you have an inorganic phosphate, a sunlight is required, and there should be an enzyme which is ATP synthase. It results in the generation of ATP. That is called as the photophosphorylation. Of course, there are types of the photophosphorylation depending upon the transport of the electrons. There are two types. One is non-cyclic photophosphorylation and another is cyclic photophosphorylation, which are accomplished through cyclic or non-cyclic electron transport system. Photophosphorylation, which involves non-cyclic electron transport system, is called as non-cyclic photophosphorylation. That means electron will move unidirectional, it will not come back. Photophosphorylation involving cyclic electron transport system is called as cyclic photophosphorylation. So first of all, we'll have to discuss the non-cyclic photophosphorylation, the mechanism of the non-cyclic photophosphorylation. It is a common process among higher plants. And as I said, the flow of excited electrons is unidirectional. We have already understood that in photosynthesis there exists two photosystems or two pigment systems. One is depending upon their absorption maxima. We have in photosystem second, photosystem 1, which are located within the thylakoid membranes. Here, two systems are involved in non-cyclic photophosphorylation. The electrons donated by pigment system second, which is also called, or photosystem second, which is called as P680 nanometers, has a reaction center P680, are not returned back. So that means electrons will be passed on from one system to another system, and electrons are carried through different electron carriers to NADP via second system, which is pigment system first, or P700. So in non-cyclic photophosphorylation, there's an involvement of two photosystems. This non-cyclic photophosphorylation is often called as Z-scheme because electron carriers, which are depicted as per the redox potential value, they assume a zigzag shape which is similar to alphabet Z. So if you will see the outline of this Z scheme, we have one system here which is called as P680, pigment system second. Then we have another system, pigment system first, P700. These are its antenna molecules, this is its reaction center. Now these two systems will accept the photon simultaneously. This will accept the photon of light, this will accept the photon of light, and once it accepts the photon of light, its reaction center gets excited the chlorophyll reaction center gets excited and electrons are passed on to primary electron acceptor, that is a pheophytin, and then it is passed on through plastoquinone A, plastoquinone B, then it goes to cytochrome Bf complex, and then electrons go down to plastocyanin, which is a copper containing protein, and then here this system, which has also accepted the photon, excites its electron to this quinone, which is accepted by primary electron acceptor, and then that electron is passed through these iron sulfur protein, ferredoxin, and ultimately in presence of FD NADPH reductase causes the reduction of NADP to NADPH plus H positive. Now this system becomes deficit. Here this system also becomes deficit of electron. Now this will transfer its electron to this system, but here when there has been the excitation of this system, that time there has also been the photolysis of the water, that is water molecule, because pigment system second contains an oxygen evolving complex. We'll discuss it separately, where at that time that photon has resulted in the release of one electron at a time and one proton, and that is given to this deficit system through a protein that is Z, and then it causes the deficit which is here in the system, so that there is a flow of electron again, and the second photon will cause the excitation of this system. As a result, you will see there is a unidirectional flow of electron, that is electron lost from this system does not come back, and that is taken ultimately by this system. And at the end you will see there is an evolution of oxygen also. This is what is named as a Z scheme. As you will see, these are the carriers as per their redox potential. They are positioned in such a way that it gives an appearance of an alphabet Z. That's why it was named as a Z scheme. Now let's understand the details of this Z scheme. And here you have seen that there is a coupling of ADP with inorganic phosphate to form an ATP. That is near cytochrome Bf complex. It was as per this scheme, but we will see the modern concept, how there is an involvement of proton driven ATP synthesis. So we have seen the outline of the scheme. So that means there is an electron flow during non-cyclic photophosphorylation between the two systems. First system is pigment system second, or P680. Upon photo excitation, it will extrude its electron. That will be picked up by primary electron acceptor, as we have seen in the scheme pheophytin. What is pheophytin? Pheophytin is a non-magnesium chlorophyll A with a redox potential value of this more than, which is value more negative than the 0.0 volts. And now we have a system which receives, this is the chlorophyll. It receives electron from water ultimately, has a redox potential of plus etna. And at the end you will see this system also results in the evolution of oxygen. Oxygen is evolved. And then protons remain in the thylakoid lumen. We will discuss this part later on, which will result in the generation of the proton motive force. So that means there is flow of electrons from pheophytin, first from pigment system second, to primary electron acceptor, pheophytin, that donates its electrons to another electron acceptor, quinone A, which gets reduced to quinone, plastoquinone, and after P680 absorbs the second photon, another electron is released, which is accepted by QB, plastoquinone, that picks up two protons from the stromal space to form plastohydroquinone, which helps ultimately in proton-driven ATP synthesis. This we will discuss in the Q cycle. The electrons are further carried through cytochrome Bf complex, as we have seen in the figure, and then reduced plastocyanin, then it is passed on to the plastocyanin as per the redox potential. And the reduced plastocyanin donates its electrons to P700, which is also located in the non-appressed portion of the thylakoid membrane, that is P700 or pigment system 1, and that has already been oxidized following the absorption of photon. As I have mentioned, two systems are simultaneously taking the two photons That is, both the systems are excited at the same time. And electrons from the pigment systems first, which are extruded, are accepted by primary electron acceptor. There are acceptors A0 with a redox potential of this much, and then there are present other phyloquinone and iron sulfur proteins, namely iron sulfur X, iron sulfur A, iron sulfur B. And then finally electrons are passed on through electron carrier ferredoxin. And from that, in presence of an flavoprotein, ferredoxin NADP oxidoreductase, that causes the reduction of NADP to NADPH plus H positive, which of course takes in protons from the stroma portion also. It is the overall flow of the electrons in the non-cyclic photophosphorylation. But we have seen there is a one component. That component is called as the photolysis of water. Because when there is an excitation of the pigment system second, excitation of the pigment system first, at the same time, the ultimate source of the electrons is the water. The water makes the cycle to run. So what we mean by the photolysis of the water, literally it means the lysis of the water in presence of the light, breakdown of the water in presence of the light, oxidation of the water in presence of the light. So light induced splitting of water into protons, electrons, hydroxyl and oxygen in presence of a water oxidizing enzyme. Tyrosine residue, 4 manganese ions, chloride ions and calcium is known as photolysis of water. This is the definition of the photolysis of the water. So electrons from water are transferred one at a time. Since we know one molecule of water will release 4 electrons, but the system is deficit of 1 electron. So electrons from water are transferred one at a time. Electrons pass on through manganese ions and Z, a tyrosine side chain on the D1 polypeptide to excited pigment system second reaction center to get reduced to P680, which is because it's a deficit of the electrons. Water oxidizing reaction releases 4 electrons, but pigment system first second can accept only 1 electron at a time, because if 4 are released, you'll see that there is a water oxidizing clock where water is oxidized in transient states. It's not done in one flash of light. So experimentally, it was found that chloroplasts illuminated with short saturated pulses of light. And it was also found that oxygen was evolved after 4th pulse. That means 4 photons are used, and at the 4th pulse, the oxygen is evolved. The conclusion from this experiment was that pigment system second lose an electron and subsequently oxidized the oxygen evolving complex 4 times in a succession to form a molecule of oxygen. Let's understand how it is taking place. Bessel-Koch in 1970s proposed a water oxidizing clock, according to which water oxidation occurs in several transient states. Suppose we have a water here which is in the S0 state. First flash of light comes, and due to that flash of light, there will be release of 1 electron, 1 proton. Of course, that 1 electron can be taken by this system through Z carrier. And then this water is changed from S0 state to S1 state. Second photon of light will come. That will change this S1 state to S2 state, releasing 1 electron and 1 proton. And then there is an S2 state which receives the third flash of light. And that third flash of light releases the 1 electron, 1 proton. And there is the S3 state which will receive the 4th flash of light. And due to 4th flash of light, there will be release of 1 electron and 1 proton. So by this time, 4 electrons and 4 protons are released one by one in succession. And that is taken by the Z to this system, which is losing electron simultaneously at when it accepts the photon of light. To make the system running, that this 1 by 1 electron is taken by this pigment system second. And once it has reached to the S4 state, that time simultaneously, that is the 4th flash ke baad, there is a simultaneous decay of this water releasing oxygen, releasing two molecules of water, releasing this oxygen, which is the reason that at the fourth flush of light there is evolution of oxygen. So this was the one component which is making the electrons flow in a non-cyclic direction, because there is a source of electrons, that is the water. That water is being oxidized as in a transient status, releasing one by one, making the system run. Let's understand how there is a ATP synthesis, because Z scheme doesn't explain. It has only shown that there is a coupling at cytochrome B complex, but let us see that there is a proton-driven ATP synthesis. As you have seen in case of oxidative phosphorylation in the mitochondria, the synthesis of ATP during photophosphorylation in chloroplast is actually proton-driven. We'll understand it. We know electron transport carriers in thylakoid membrane participate in both electron transfer and proton translocation. We have seen in thylakoid membrane there are present the electron transport carriers, both systems are present there, and they are responsible for electron transfer as well as the proton translocation. Protons are translocated from stroma of chloroplast to the thylakoid lumen. And then the accumulation of protons, hydrogen ions, protons within the thylakoid lumen establish a proton gradient across the membrane. So there will be proton gradient, a proton concentration will be established there across the membrane. Of course, that will help in the synthesis of the ATP also. So as a result, you'll see the thylakoid lumen has more acidic pH than stroma. It generates an electrochemical potential difference across the membrane. So there is a difference on the two sides of the membrane. That's called as the electrochemical potential difference. Now you will see when there is as a consequence, if the protons are more within the lumen side of this thylakoid, there will be an efflux of protons as per the concentration gradient through an ATP synthase CF0, CF1 particle, which helps in, that's a ATP synthase, which helps in coupling of ADP with inorganic phosphate. That results in the synthesis of the ATP. This concept at least we have seen in oxidative phosphorylation when we have also gone through the chemiosmotic hypothesis proposed by the Peter Michel. We will explain that hypothesis here also, because it also explains the ATP synthesis in the chloroplast. Let us explain with this diagram. This is the cross section of the thylakoid. This is the thylakoid membrane. This is the thylakoid lumen. This is the membrane where we have the electron, we have the proton pigment system second, and we have pigment system first. And these are the carriers which are connected, as we have seen in case of Z scheme. This is the pigment system first, there is iron sulfur, FD ferredoxin, and then this is on the outside, that is towards the stroma side. So we have a thylakoid membrane where there are present where there are present electron transport carriers, and there is also present this which is called as ATP synthase, which has a CF0 and the CF1 particle and has a proton channel through it. So we have this system. Towards its lumen side, there will be photolysis of water, which will result in the generation of protons as well as the electrons. So more protons will be towards the lumen side. And then there will be the transfer of electrons to PHE to plastoquinone pool, and we will see this Q cycle as we have already discussed. We will give the summary of it here also. This Q cycle and this cytochrome BF complex is ultimately responsible for the translocation of protons towards the lumen side from stroma side. And so we have more protons towards the lumen side than towards the, so across this membrane, there is a potential difference on this side as well as on the stroma side. Ultimately, electrons are passed from this to a mobile carrier, plastocyanin, that passes on to this system, which also accepts the photon of light, extrudes its electrons, and that electron is given by this system. So there is a non-cyclic electron flow from this, but ultimate source is the water here. So we will try to understand that then there is a formation of NADP gets reduced to NADPH, that is towards the stroma side. And here we have now more protons. On so as the sides of the stroma of the thylakoid, there is less proton gradient. There will be a movement of protons from this through this, which will result in the coupling of ADP with inorganic phosphate. As a result, there will be the formation of an ATP, which is also called as the assimilatory power. So this will result in the generation of ATP. Also, there will be the reduction of NADP to NADPH. Together, we call it as an assimilatory power. So this is how proton-driven ATP synthesis is taking place in the chloroplast thylakoid. Let's understand the different components of this, especially the Q cycle and how it is taking place here. What is Q cycle? As we have given a detailed lecture on the Q cycle, please refer to that. We have seen that proton translocation occurs through Q cycle. Q cycle is a cyclic process involving oxidation of plastohydroquinone, and there is a differential movement of two electrons. One passing through a linear chain comprised of iron sulfur, cytochrome F, and then it goes to the plastocyanin mobile carrier towards the pigment system first. Others through two B-type cytochromes to reduce semiquinone to plastohydroquinone. It helps in movement of protons from stroma to thylakoid lumen. The Q cycle has a role. It will help in the generation of the proton gradient towards the lumen side. There is a net oxidation of one plastohydroquinone to ubiquinone or plastoquinone, and two electrons proceed to cytochrome C on their way to oxygen. Let us see the summary of the Q cycle, because it's discussed in two halves. One is the first half of the Q cycle. This is the thylakoid. This is the thylakoid. This is one system, that's pigment system second. This is the pigment system first, located within the thylakoid membrane. And this side is the lumen side. This side is the stroma side. So we have a stroma side here, we have a lumen side here. We know how pigment system second is responsible for the Q pole generation. The way the quinone will be changed into semiquinone, then plastoquinone, and then how plastoquinone will form the plastohydroquinone by accepting the protons from this side. Presuming that within the Q pole there is already the generation of plastohydroquinone. We have a cytochrome BF complex. As we have discussed, this cytochrome BF complex has iron sulfur rich key proteins, cytochrome F, that is a heme C. Then there is present cytochrome B6, which is comprised of B, cytochrome BP, cytochrome BN, and this cytochrome has two sites, catalytic sites. One is called as the QI site, which is called as input site, and QO site, which is called as the quinone output site. Now what's happening, briefly I'm trying to explain it here, that once there is a plastohydroquinone, if one plastohydroquinone is taken by this Q site, QO site, output site, there is a differential transference of electrons. One electron will move through this direction from this BP to Bn. Another will pass through a linear chain, that is called as, it passes through iron sulfur protein, then to cytochrome, and then it will be taken by the PC, plastocyanin, to be transferred to the pigment system first. At the same time, it has passed on one electron to this system, but one electron is here, where we will find there is a plastoquinone that will be accepted by the quinone input site, where this will take, quinone will be accepted at quinone input site, where electron will be taken by that, and it will accept one proton to become the plastoquinone. Now this is the first half of the cycle. One electron has passed to this. One is locked here. What will happen in the second half of the cycle? Second half of the cycle, from this pool, another plastohydroquinone will come here. And there will be now, and at the same time here, what has happened, I have forgotten here, there is translocation of 2 protons. Because this plastohydroquinone, it has lost 2 electrons, it will pass on 2 protons towards the lumen side. Now second has come here. Second one passes on electrons to 1 electron differential movement of the electrons. One will move to this direction and one will move to the linear direction. That is from this iron sulfur and cytochrome F and then to plastocyanin. And at the same time, it will translocate 2 more protons. But 1 electron is carried through cytochrome B6 through these 2 and is accepted by this semiquinone. Because 2 electrons will be there, now it will accept 1 more proton there to regenerate plastohydroquinone. So if there is a utilization of 2 plastohydroquinone, there is a regeneration of 1 plastohydroquinone which goes to the Q pool. I hope you have understood how a Q cycle, that's why we have seen in Z scheme, there is a the coupling of ADP with inorganic phosphate to form an ATP has been shown there. But it is in relation to this Q cycle because it helps in the translocation of the protons. 2 protons here, then 2 protons here, that is 4 protons are translocated from stroma side towards the lumen side. So we have understood that there is a Q pool and that is responsible for operation of this Q cycle. So overall, we have seen that 4 protons are transferred across the membrane for every 2 electrons delivered to system. Very important point. 4 protons are transferred. You have seen it in Q cycle. 2 in the first half, 2 in the second half. And 2 protons for every 2 electrons delivered to the pigment system. So 2 more protons are released in the lumen for each molecule of water. That is oxidized to oxygen during photolysis. We have already seen it of water. Further, 1 additional proton is removed from the stroma. That is for the reduction of each NADP to NADPH2. Thus, electron transport performs the osmotic work of accumulating proton, generating a pH difference across the membrane. So what is the role of the electron transport? That is to generate a potential difference across the membrane. See, we'll see the sum up of the how many protons are accumulated within the thylakoid membrane. If you visit the summary of it, proton accumulation in thylakoid lumen, 1 mole of oxygen, 2 water molecules, there will be release of oxygen molecule, there will be release of 4 protons, there will be 4 electrons. And if 2 electrons, that will result in the generation of 4 protons. As we have understood, when 2 electrons are passed, 4 protons are translocated. Since we have 4 electrons here, so that means there will be 8 protons. So overall, a water molecule, 2 water molecules will result in the formation of an oxygen, and there will be 12 protons translocated towards the thylakoid lumen. Overall photosynthesis, which is 6 times carbon dioxide, 12 times water, releases glucose plus 6 times oxygen plus 6 times water. If you will see the budgeting of the accumulation of the protons, 1 mole of oxygen we know 12, 6 moles will be 6 into 12, 72 protons. So we have understood that 4 proton is required for ATP molecule. So therefore, that is 72 into 4, that is 18 ATP are generated there, as you will see. So 12 water molecules are there. That will result in the accumulation of 24 protons, 24 electrons, and 6 water molecules will be released back. So what a Q cycle is doing, 1 pair of electron, 4 protons translocate towards the thylakoid lumen, 12 pairs, that will be 48. Total protons towards thylakoid lumen will be 24 plus 48, that is 72 protons. 72 protons into 4. That is 4 are required for this. So there will be 18 ATP synthesizer. This is the overall accumulation of protons because of the this Q cycle, which is responsible for accumulation of protons towards the thylakoid lumen, which creates the proton gradient. We have seen a similarity exists between photophosphorylation and the oxidative phosphorylation or respiratory phosphorylation. So both are working on chemiosmotic mechanism proposed by the Peter Michel, and we have also seen that there is a structural similarity existing between the ATP synthase here as well as in case of the mitochondria. Let us see what are the effects of herbicides on this electron transport system or on this photophosphorylation. Non-cyclic photophosphorylation and evolution of oxygen is inhibited by two herbicides, rather three herbicides, namely DCMU, dichlorophenyl dimethylurea, diuron, and paraquat methyl viologen. What are they doing? We'll see it in the diagram. They are blocking the electron flow. At this DCMU is blocking the electron flow at the quinone acceptors of pigment system second by competing for the binding site of plasstoquinone between QA and QB. Diuron picks up the electrons from early acceptors of pigment system first and reacts with oxygen to form superoxides of oxygen. And that superoxides of oxygen damages the lipids of the chloroplast membrane. So the herbicide works on making this electron transport system weak so that there is no generation of assimilatory power within these herbs, within these weeds. And paraquat accepts electrons from the ferredoxin of pigment system first and transfers them to molecular oxygen, forming the reactive oxygen species. And that reactive oxygen species or the superoxides of oxygen will result in the damages of the lipids of the chloroplast membranes. This is how it works. We have this pigment system second, pigment system first. These are its carriers. This is its carrier. And we know the electrons are passed on through this. Here is the DCMU. DCMU competes with this QA. So once it is, this chemical is here, the DCMU, diuron also is here, it will compete there, so there will be no translocation of the electrons. Once there is no electrons, there will be no generation of ATP and the system will not work. Likewise, the paraquat or the methyl viologen blocks here ferredoxin. It blocks the electrons here and reacts with the oxygen, forming a reactive species of the oxygen that will damage the lipids of the chloroplast membrane. So there will be no photosynthesis. Once there is no generation of the assimilatory power, the plant turns weak, and that results, that's the basic funda behind the herbicides. What is the experimental proof or the direct proof towards the photophosphorylation? It was the first experiment which was done by the Henry Jejendorf and its co-worker Uraib. They gave a first experimental proof in support of a chemiosmotic mechanism of photosynthetic ATP formation. They demonstrated that establishment of an electrochemical gradient across the thylakoid membrane could lead to ATP synthesis under direct conditions. They established an artificial proton gradient in absence of light. That can be done. They took an isolated chloroplast and kept in buffer pH 4 and then kept there for a few seconds, 60 seconds, and then to equilibrate it, and they transferred it to another beaker which was having the pH 4, same pH, so that it gets equilibrated. And then these thylakoids which were kept in the pH were simultaneously transferred, rapidly transferred to the pH 8 buffer, and thereby creating a pH difference of 4 units across the thylakoid membrane. The inner of it is having the pH 4, the outer of it is having the pH 8. There's a difference across the membrane. And then by simultaneous in presence of ADP and isotope labeled inorganic phosphate P32 under complete dark conditions, that led to ATP synthesis. So they suggested that actual role of light in chloroplast during photochemical phase is to create a proton gradient. Since they established the proton gradient here, artificial proton gradient, and adding an ADP and inorganic phosphate for few seconds, they were in position to generate ATP. This was done by Andrei Jajenderov, a plant biologist who is notable for providing direct evidence that chloroplasts synthesize ATP. This is the experiment. They isolated the thylakoids and put them in pH 4. After equilibration for 60 seconds, then they transferred it to another which is having a pH 8. So there was a potential difference between the two, and then keeping it in a dock, adding the ADP and labeled inorganic phosphate, there was the generation of ATP. This was the direct proof towards the photophosphorylation or proton-driven ATP synthesis in the chloroplasts. So we have one more photophosphorylation, which is called as a cyclic. So far we have seen the non-cyclic photophosphorylation. Here in cyclic photophosphorylation, there will be, as the name signifies, there will be the return of the electron. Flow of electron is directional, involving only pigment system first, that is P700. It does not involve the two systems. The electrons expelled by reaction center P700 returns back to it after passing through electron carriers of an electron transport system. It is of limited occurrence in higher plants, especially under low light intensity and reduced carbon dioxide supply. So you will see no oxygen is involved because oxygen evolving complex is present in the pigment system second. So it's only involving the pigment system first. So no oxygen is involved here. And there will be no reduction of NADP here. Also, it is prevalent in anaerobic photosynthetic bacteria, chromatium, and it supplements ATP production for dark reaction where there is an excessive accumulation of NADPH plus H positive. It also acts as a source of additional energy to drive other endergonic metabolic processes. It can retard rates of carbon dioxide reduction as no NADPH is produced. A source of ATP, especially in bundle sheath cell chloroplasts of C4 plants. It is the significance of cyclic photophosphorylation as it is also taking place in several plants under certain conditions. How it is taking place? We have a pigment system. First, P700, these are antenna molecule, accepts a photon and its electron is excited to this primary, then passing through these carriers, which are of course present in the non-cyclic photophosphorylation also. It uses these carriers, A1, iron sulfur X, iron sulfur SB, iron sulfur SA, then ferredoxin, and then goes to the plastoquinone. From plastoquinone, cytochrome BF complex, and then to the cytochrome BF complex to electron is passed to PC, and then it goes back to the system. So there is a directional movement of the electrons. That's why it is called as the cyclic photophosphorylation. It of course also shows here there in presence of an ATP synthase. There is a coupling of ADP with inorganic phosphate to form ATP. So ATP is also generated, but no NADPH reduction takes place here. So this is the another view of it is as the energetics of the cyclic electron flow, how this system works here. Then it goes through these carriers, then to ferredoxin, and then there is a cytochrome, and then there are proton transport. Then this is the PC back to this as per the free energy, as per the redox potential. And on absorbing photon, let us give the description of it is. On absorbing photon of light, P700 becomes excited. This high energy electron is expelled, leaving chlorophyll molecule in oxidized state. Electrons will tend to flow from state of higher to the lower free energy, from more negative to more positive redox potential. The expelled electron is picked up by special chlorophyll A monomeric molecule AO and is passed over to carriers phyloquinone A1, a derivative of vitamin K. And then there are series of these carriers, and then to ferredoxin, and then to plastoquinone, and then plastoquinone helps us in the picking of the protons and translocate the same to the thylakoid lumen. And same proton-driven ATP synthesis also takes place here. Translocation of proton into thylakoid lumen generates proton motive force across the thylakoid membrane. Proton motive force is utilized for ATP synthesis. Electrons from plastoquinone are passed on to cytochrome BF complex, which in turn hands over the electrons to oxidized P700 via mobile or soluble carrier plastocyanin. This is how the description of it is taking place. And then we will see how creation of proton motive force favors efflux of protons through proton channel of F0 of ATP synthesis that results in the coupling of ADP with inorganic phosphate. This is how it is taking place here. We have seen this pigment system. This is the thylakoid membrane. This is the lumen side. This is the stroma side, and the photon is accepted here. It's passed through these electron carriers, and then ferredoxin, and then plastoquinone. Plastoquinone helps in the translocation of protons from stroma side towards the lumen side. And since towards the lumen side there are more protons, and that proton will, as per the gradient, there will be an efflux of the protons from this through this ATP synthase and F0, F1 particles, which will result in the coupling of the ADP with inorganic phosphate to form an ATP. So this is how proton-driven ATP synthesis also takes place in the non-cyclic photophosphorylation. So in totality, we have seen here photophosphorylation, its definition, its mechanism of working, the role of Q cycle, photolysis of water, and then how there is a proton-driven ATP synthesis involved in the chloroplasts and some direct evidences in it. I hope you have understood the mechanism of the photophosphorylation. Hope you have been benefited by this video, and see you in the next video. We will be discussing the other aspects of the photosynthesis. Till then, be safe. Thank you for patient listening.